Hi everyone, am I audible? Hello. Hi everyone, am I audible to you all? Hi everyone, am I audible to you all? Happy morning, welcome to Unacademy NEET English. I am your biology educator Ambika Sharma, and again today it is the lecture number 4 on cell chapter of course, and today we are going to talk about the cell wall, we are going to talk about the endomembrane system. So how are you all? Tell me, how are you all? Good morning, good morning. So how's this timing? 9 a.m. It's good na? In the morning you people revise the biology, then you can in the morning we read the biology and you can revise it throughout. Very good. It's better. I'm good. Thank you so much for asking. Okay, so tell me, did you solve any question from the last topic? Any question? Are you practicing the PYQ or any other MCQs? Good morning, good morning, good morning. Excited for the class? Very good. You should be excited. Even I am excited for your class. Because of this class, even my routine is getting better. I'm waking up early in the morning, right? Then I used to check what I'm going to teach you now, right? And I plan for the entire for my day. So it's good. To get up in the morning is good. Hannah? So see, if you have just started your NEET preparation, so this is my advice. You should follow this routine that like you can get up early in the morning, then you can study, you can use your entire day. But it's okay if you are even studying at night, it's fine. But right now, if you can work on this routine, that will be very, very, very good. Okay, that will be very, very, very good. Okay. Done. So today let's start talking about the cell wall. So if you have any doubt from the last topics that we have covered, do let me know. And please kids, don't be like that, ma'am, why are you taking 4 or 5? See, our first lecture was a kind of waste because in that lecture, because of some settings, I was not able to teach you properly, right? So you can consider it as your third lecture. And don't be like that, ma'am, why are you dragging this chapter so much? Right now, our focus should be quality content, isn't it? Isn't it? We need to cover the topic and we need to understand each and everything from that particular topic. Okay. So right now we have the time, we should cover our syllabus properly and then we can revise. We should not leave anything right now. Okay. So that is why we are going to discuss the things properly, and whatever you can say that if it is going to take 6 lectures, I'm going to take that. But right now my agenda is to provide you the quality content, that's all, because we are on YouTube, we are going to cover the high weightage chapters only. Okay, so now let's talk about the cell wall. So kids, you know about the cell wall, isn't it? Remember Sean, who was a zoologist, but he was the one who checked this, who noticed that in the case of plants, there is one more outer covering which is not there in the animal cell. And that outer covering is what? That outer covering is what? That is the cell wall. That is the cell wall, right, bache? So now when you talk about the cell wall, how to discuss that? See, I'm again going to relate it with the different kingdoms. So you know that we have Monera, we have Protista, we have fungi, we have Plantae, and then we have Animalia, isn't it? Isn't it? These are the 5 kingdoms that we have, right? You're not going to forget it. Whenever I'll start biological classification now, right, you will find it useful. So now in the Monera we have the bacteria, and in the case of bacteria also we have the cell wall. So now everyone in the chat section, do let me know what is present in the bacterial cell wall, right? What is present in the bacterial cell wall? Do let me know. Now comes the Protista. Here in Protista, some organisms are having cell wall and some may lack cell wall also. Now in the fungi also cell wall is present. In the plants also cell wall is present, but it is absent in the case of animal cell. In the case of animal cell, there is no cell wall, right? There is no cell wall. So as I said, in the case of bacteria, what will be there in the cell wall? We have already covered it. There will be the peptidoglycans, and we also call it as murines, and we also call it as murines. Are you getting my point? So along with these peptidoglycans, some short peptides are also present here in this bacteria. Okay, some short cross peptides, some peptides, short peptides, some proteins are also there in this bacteria. Right, bache? And very good, Satish, you are right. When it comes to the peptidoglycans, we have NAM and NAG. We have N-acetyl-muramic acid, we have N-acetyl-glucosamine. N-acetyl-muramic acid, N-acetyl-glucosamine. Now, when it comes to the fungus, so can you tell me what do you have in the fungi cell wall? Yes, anyone? What do we have in the fungi cell wall? When we talk about the fungus, bache, in the fungi cell wall, we have chitin. What do we have? We have chitin. Again, a repeated MCQ. Okay, again it is a repeated MCQ. What do we have? We have chitin, and chitin is a homopolysaccharide, bache. Chitin is what? It's a homopolysaccharide. Chitin, homopolysaccharide. We even call it as fungal cellulose, right? We even call it as fungal cellulose because it is present in the fungi. And what is the meaning of this term? That it is homopolysaccharide. Homo means same. Same. Polysaccharide, having so many sugars, right? Having so many sugars. So when it is the homopolysaccharide, it is again made up of NAG. So many N-acetyl-glucosamine, they will join, they will join, they will join, they will form the chitin. Again, it is a previous year question. Do not consider it as something extra, bache. It is again given in your NCERT. It's just that I am putting everything here in the same topic. Okay, these lines are even mentioned in the NCERT. So this is what you need to remember. The chitin, which is a fungal cellulose, it is present in the fungi cellulose, fungi cell wall. It's a homopolysaccharide made up of N-acetyl-glucosamine, made up of so many N-acetyl-glucosamine. This is the first point. And moreover, this chitin is even present in the exoskeleton of insects. Do you remember this? Chitin, even it is present in the exoskeleton of insects. Right, like cockroach. Okay, their exoskeleton is also having the chitin. So it is a repeated question that you need to remember. Now what are we going to study? We are going to talk about the plants. We are going to talk about the cell wall of the kingdom Plantae. Okay, right. So in the case of plants, algae, bryo, pterido, gymno, angiosperms, all are present. Right, bache? When you talk about the algae, along with cellulose, in the algae, we have some other compounds also. But as of now, let's talk about in general, let's talk about the plant cell wall, and then individually we are going to mark that things from the NCERT. Right, bache? So when it comes to the cell wall, the outermost covering in the case of plant cells, like let's say this is the plasma membrane in a plant cell, and here we will be having the cell wall. Now bache, when you talk about the plants, you know that they are having the large vacuole, isn't it? They are having the large vacuole, and in the case of plants, the cell wall is very important. It is going to maintain the shape. And do you know how the plant cells grow? Basically, the water will enter here, right? The plant cell will elongate. So cell wall will maintain that shape. Cell wall will help in that. Okay, cell wall will help in that. So this is what you need to remember. In the case of plant cell wall is very important. It is permeable. It will allow the entry and exit of things. It is permeable. And even the cell wall, it plays role in fighting with the infection as well. So cell wall is going to provide the protection, of course. It is going to provide the shape, of course. It is going to help in the cell elongation, yes. And it is going to fight with the infection, yes. So this is the role of cell wall in the case of plant cell. So Akhilesh, even it is your homework as well. Right, you people need to mention it in the comment section that what is the role of cell wall in the case of plants. I mentioned few points, 2 to 3 points. Rest you people are going to write, rest you people are going to mention. So are you guys going to do that? Yes? Will you do it? You should. Okay, you should. Done, bache? So this is about the cell wall. So now when you talk about the cell wall, the composition is, in the cell wall we will be having the cellulose, even hemicellulose will also be there. Okay, and even the pectin is present in the cell wall. So what is the composition of cell wall? In the cell wall, cellulose will be there, hemicellulose will be there, pectin will be there. and one by one we will study about the primary cell wall, about the secondary cell wall. So you will see that in the secondary cell wall further other depositions are also there, like lignin, right? All that things are also present. Clear bache? All that things are also present. So mainly in the plant cell wall, what will you get? You will get cellulose, hemicellulose, pectin. Cellulose, hemicellulose, pectin. Cellulose, hemicellulose, pectin. This is what you people are going to get in the plant cell wall. Is that clear? Now bache, when we talk about this cellulose, cellulose is also a polysaccharide, right? It is also a polysaccharide. Saccharide means sugar, poly means many. So basically it is a carbohydrate. The carbohydrate which is made up of many monosaccharides. Right? It is a carbohydrate which is made up of many monosaccharides. What is it? It is cellulose. It is cellulose. So now bache, when you talk about the cellulose, again it's a polysaccharide and it is made up of glucose. Glucose is the monomer here. It's a branched polysaccharide. Cellulose is unbranched polysaccharide. Right? So it is made up of glucose, and bache, let me tell you, this is from the biomolecule chapter. It is a beta polysaccharide. It is having beta 1,4 linkage. You can mention it in your notes. It is having the beta 1,4 linkage. I think you all are aware that we cannot digest the grass. Do you know that? We cannot digest the grass. Right? Right? In our body, we do not have the enzymes that can digest the grass. If you are going to eat the grass, obviously it will not be digested in your body. You have to cook it, right? You have to cook it semi or full. You need to cook it first. You cannot digest it raw. What is the reason behind? We do not have the enzyme cellulase. Yeah, we do not have any enzyme that can act on beta polysaccharide. Like in our diet, we used to have starch, right? Starch is also a polysaccharide. In our diet, we are also having the starch. Starch is also a polysaccharide, right? But in our saliva, like carbohydrate digestion, it starts in saliva, na? So in the saliva, we have the alpha salivary amylase, right? This is the word that we used to mention. In the saliva, we have the alpha amylase. Our amylase is alpha, right? It is going to break that polysaccharides having alpha bonding, right? Having alpha bonding. So alpha beta is nothing. It is just related to the position of OH and H. OH and H, OH and H. This is what you people need to remember. Clear bache? So in our diet, because the cellulose is having beta linkage, right? So because of that, we do not have any enzyme which is going to act on beta carbohydrates. So we are not able to digest it. Okay. So this is one of the aspect of cellulose. I think you should remember it. Even we are going to discuss it in the chapter digestion and absorption, right bache? So what I just said, that cellulose is a polysaccharide made up of glucose and it is unbranched. Along with that, we have hemicellulose also, we have pectin also, right? So these are the things which are going to form the cell wall. Now bache, one more point is there. Can you tell me who's going to help in the formation of the cell wall? Like in which organelle the component of cell wall will synthesize? Anyone? Can you tell me in which organelle of the cell the components of the cell wall will be synthesized? Tell me. Tell me, anyone. Do you have any idea? Do you have any idea? Yes? Gokul, how come mesosome? We are talking about the plant cell wall here. Yes? Anyone? How come mesosome in plant cell wall? Seriously, plasma membrane, I am talking about the organelle. I have mentioned it, I have specified it. I am talking about the organelle. Seriously, that's very bad, bacho. When you talk about the plant cell wall, the cellulose will be synthesized, right? It will be synthesized in cell membrane with the help of enzyme. It will be synthesized in cell membrane, or you can say that it will be synthesized by cell membrane, right? Cellulose. But when you talk about hemicellulose and pectin, obviously in the Golgi apparatus, right? In the Golgi apparatus it will be. In the Golgi apparatus it will be synthesized. Okay, so it is a repeated PYQ, that which cell organelle is going to help in synthesizing the plant cell wall. So it is the Golgi apparatus, and in the case of plants we also use the word dictyosome for it. right? In the case of plants, we also use the word dictyosome for it, for the Golgi body, okay, for the Golgi apparatus. But when you talk about the cellulose, it will be synthesized by cell membrane, because the enzyme that is going to synthesize the cellulose, it is present in the cell membrane. Cellulose synthase, it is present in the cell membrane. So this is what you people need to take care. This is important. This is important, and it is one of the functions of this Golgi apparatus, bache. Right? How can you say that mitochondria? And how can you specify mesosome in the case of plant cell wall? In the case of plants, we do not have mesosomes. We talk about the mesosomes in the case of bacteria, bache. What are you doing? Don't take this chapter casually. Right? You never know. Maybe this year, no doubt the biology was very easy, but next year, anything can happen. You cannot predict your paper that next year also it is going to be very easy. Right? We should write a note to NTA that these days the level of paper is not so good. And we want good doctors. Literally, we want good doctors. Okay, so this is what you need to remember. So cellulose will be synthesized by cell membrane, because the enzyme is present in the cell membrane, and when it comes to hemicellulose and pectin, it will be synthesized in the Golgi apparatus, right? It will be synthesized in the Golgi apparatus. Now the next part. So when you talk about the cell, okay, so when you talk about the cell wall, there are two things, right? There are two things. The first is primary cell wall, and the second is secondary cell wall. Firstly, let's discuss that. Primary cell wall, and second is secondary cell wall, right, bache? So when you talk about this primary cell wall, it is thin, it is elastic, and it is even living, okay? And when you talk about the secondary cell wall, it is obviously thick, and it is considered as dead. It is considered as dead, right, bache? And when it comes to the primary cell wall, here the growth, when it comes to the growth of primary cell wall, it will be intussusception. I'll explain all the words here, and here the growth is going to be accretion. Now let me explain this part. Now when you talk about the plant cell, let's say here we have one plant cell, and here we have another plant cell. Now bache, in the case of plant cells, we are also having a kind of cementing layer in between, right? In the case of plant cell, we are also having a cementing layer in between. Right? We are having what? We are having a kind of cementing layer in between. Here you guys can see. See, let's say this is that cementing layer, but it is important. It is going to join two plant cells together, right? We are talking about what? We are talking about the plant cells. Now just focus here, na? The diagram is important. Just have a look, it is important. Now, just look at this diagram. Now here, this innermost is your cell membrane. Okay, selectively permeable cell membrane. Now these layers, this one, it is basically this layer is basically the secondary cell wall, right? And then we have this orange one, it is the primary cell wall, right, bache? It is the primary cell wall, and this white colored layer is middle lamella. What is it, bache? It is middle lamella. Now I'll explain it. See, in these two cells, this cementing layer is middle lamella. It is present in the plants. What is it? It is the middle lamella which is present in the plants. Now bache, it is something very important. This middle lamella is made up of calcium and magnesium pectate, right? It is having calcium pectate or magnesium pectate. Its composition is important and it is a cementing layer. Now when you just look at this, just look at the sequence of the layer, right? Outermost is middle lamella, then comes the primary cell wall, then comes the secondary, and finally there is the plasma membrane. Usually what students used to think, that there will be the primary, there will be the cell membrane, then primary, then secondary. No, it is not like that. Outermost is middle lamella, and after that middle lamella you are having the primary cell wall, then there will be the layers of secondary cell wall. Primary cell wall is is single, right? There is just one primary cell wall, and then you guys are having the secondary cell wall. You people are having the secondary cell wall, and innermost is the cell membrane. So it is also one of the questions asked in previous year paper. So you should know about this sequence, right, bache? You should know about the sequence. So middle lamella, primary cell wall, then secondary, and then cell membrane. Clear, bache? From outside to inside, this is the sequence. Now when you just look at these two cells, here in middle lamella I am showing this gap now. Actually, bache, this is a continuous cytoplasmic connection. This gap that I am talking about here now, it is the continuous cytoplasmic connection, and we used to call it as plasmodesmata. So P for plant, P for plasmodesmata, right? P for plant, P for plasmodesmata. So what is it? It is a continuous cytoplasmic connection, right? It's a kind of gap junction in the case of plants. So in the case of plants, the name for that gap junction is plasmodesmata. The name for that gap junction is plasmodesmata. Now bache, when it is a primary cell wall, now when you talk about the thin, just a minute, when you talk about the actively dividing cells, can you quote any example of actively dividing cell in the case of plants? Yes, bache? Can you just mention any single example of actively dividing cell in the case of plants? Very easy it is. Do you remember that? Actively dividing cell, like meristematic cell. So when you talk about such cells, they are small in size, and in their case, because they want growth, they are even small in size. So in such cells you will only find the primary cell wall. The only primary cell wall will be there. When a cell is small, when it is growing, when it is actively dividing, at that time only this thin primary cell wall will be present. Right, at that time only thin primary cell wall will be present, and even such cells will be having dense cytoplasm, the large nucleus will be there. But here the main point is, when the cell is actively dividing, it is growing, the cell wall is going to be very thin, and it is the primary cell wall. It is the primary cell wall. Are you getting my point? Now we consider this cell wall as living, because when the cell wall is present, the cell is capable of growth. It is growing. Okay, it is growing. Are you getting my point? It is growing. Are you getting my point? Right? So in the case of primary cell wall, the growth is, like in the case of living organism, the growth is intussusception. What is the meaning of intussusception? Intussusception means internal growth. Right? Intussusception means internal growth will be there. Right, we are the living organisms. Internally we are growing. Like let's say there is a layer, right? So when it will grow, it will become like this. Internally the material will be added to this layer, right? Material will be added to this layer. So this is about the primary cell wall. Okay, right? This is about the primary cell wall. Internal growth, intussusception is the word here. It will help the cell to grow. Now let's talk about the secondary cell wall. So secondary cell wall will be there in a mature cell. When the plant cell is mature, it will have the secondary cell wall, and we consider it as dead. Let me tell you the reason. Now see, as I said, here you have the primary cell wall. Now when it comes to the secondary cell wall, it will form in the layers. Right, bache? It is the basics. These are the basics basically that we used to even study in the chapter living world. Right, that we used to study even in the chapter living world. Are you getting my point? Right, so what is happening? Secondary cell wall, mature. It will be present in the mature cell wall. It will be thick. It is a thick layer. So when a cell will have secondary cell wall, it will not show the further growth. Right, the further elongation will not be there in that particular plant cell. So you can see here, bache, you can just have a look. So it will be in the form of layers. Even if you will check the NCERT, in the NCERT it is mentioned S1, S2, S3. Right, so deposition, this will grow with the help of deposition. Like I'll give you the example of a non-living thing. Right, we used to say now that growth is the feature of, no doubt growth is the feature of living organism, but again we see the growth in the non-living organisms as well. We even see the growth in the non-living organisms as well. Like let's say there is a stone. So over the stone, you know that many layers of over the stone, you can say that many layers of soil can get deposited. Obviously it will increase in its size. With the time it can happen, isn't it? Isn't it? With the time it can happen. Like let's say there is a field, the stone was placed there. So if you will check even after a month, you will see a kind of layer will get deposited over it, right? A kind of layer will get accumulated over it. So that is also a kind of growth. But for such growth, we use the word accretion, right? We use the word accretion. And A for accretion, A for accumulation. This is how you can relate it and remember it. A for accretion, A for accumulation. So in this case, what is going to happen? There is one layer, over it the another layer will get deposited, over it the another layer will get deposited, and this is how it is going to increase its size. So in the secondary cell wall, we have such growth where the layers are going to form, right? Where the layers are going to form. But primary cell wall, living it is, intussusception will be there in the case of primary cell wall. Are you getting my point? So up to this part, all clear? So that is how the things are present, Anuradha. That is how the things are present. So here we can see middle lamella made up of calcium and magnesium pectate. Then comes your primary cell wall, which is living, showing internal growth, which is not so thick, which is thin, which is capable of elongation. Then comes the secondary cell wall, which we will see in the case of mature cell, right, which will not show further growth. And when it comes to the secondary cell wall, it will be in the form of layers. It will be in the form of layers. So S1, S2, S3 like this. And then comes the plasma membrane. Okay, then comes the plasma membrane. Clear, bache? Clear, bache? The position is clear to you. Now, in some cases, you can even find the tertiary cell wall. In the case of gymnosperms, you people will even see the tertiary cell wall. Tertiary cell wall. Obviously, it will be internal to secondary cell wall. Okay, like here you have middle lamella, then comes primary cell wall, then comes secondary cell wall, and then the tertiary cell wall. Okay, and finally there will be the plasma membrane. Got it? There will be the plasma membrane. So that is how you need to remember this. Now, bache, when you talk about this cellulose, see what is happening in the case of cellulose. This word is given in NCERT. First of all, so many glucose in thousands, so many glucose together, right? They are already forming the cellulose. Now that so many glucose, right, they are going to form the bundles. Right, they are going to form the bundles. Like let's say here you are having so many glucose together. They are going to form the bundles, right? And for that bundles, we use the word microfibrils. So in the case of plant cell, we use this word microfibrils of cellulose. So ultimately, they are the bundles of glucose. They are going to provide the strength. So when the cellulose, which is a polysaccharide, together when it forms the bundle, right, it is forming the microfibrils. So it is going to provide strength to that cell wall, right? At this time, it is going to be very strong. It is going to be very strong. So it is going to provide the strength, bache. It is going to provide the strength. And when you talk about this pectin is a negatively charged. So what will it do? It is going to trap positively charged particle. When you talk about the pectin, it is negatively charged and it is going to trap some positively charged ion. Like pectin, which is present in cell wall, it can even trap calcium ions. And because of that, what is happening? It will provide it a jelly-like consistency. Right? It will make it a gel. The pectin is negatively charged, going to trap some calcium or something, right? Then it will also hold water. So it will provide some gel-like consistency. Some gel-like consistency, this pectin. So even this pectin is used to make the jams, to make the jellies. Okay, it is extracted and it is used to make the jams, jellies. Are you getting my point? Pectin. Okay, so in the case of leaves, in the case of fruits, you will not find secondary cell wall, but otherwise in the bark there will be the secondary cell wall. That is why we used to say that when Robert Hooke, he observed the bark of the tree, he just saw the secondary cell wall. He just saw the dead cell. Okay, done, bache? Is that clear? Is that clear? So remember this, negatively charged going to trap the positively charged ion, which will hold the water then, and it will make the gel like consistency. So it is used to make the fruit jams and the jellies, and moreover it fights with the infection as well. Okay, so in the cell wall, this is the pectin. It is the pectin which is going to fight with the infection, So this is what you people need to remember. So here you guys can see the arrangement as well. The outermost is the middle lamella, bache, right? Outermost is the middle lamella, made up of calcium pectate and even the magnesium pectate. Then comes the primary cell wall, then secondary, and then finally there will be the plasma membrane. And here you guys can see the cellulose microfibril. So basically these are the bundles. So in between the pectin is present, in between even the hemicellulose is present. Okay. So in some cells, some other deposition, lignin, fibrin, even such things are also present. Have a look, bache. See this primary cell wall. Have a look of this primary cell wall. Right? How is it growing? The plasma membrane, the primary cell wall, right, bache? And then see the secondary cell wall, which is in the form of layers. S1, S2, S3. So many layers can be there. So see this. So even in this secondary cell wall, we can have other depositions also, like lignin can be there, right? And some proteins are also present. Here also protein is there, the pectin is there, and even see cellulose microfibrils are there. Okay, so you will get these notes in the Telegram group. So do join the Telegram, bache. I will provide you everything there. So see one more diagram. So secondary cell wall, S1, S2, S3, primary cell wall and middle lamella. I hope it is clear to you. Is that clear? Sure, bache? Sure? Okay, so see the genes are going to form the protein. That proteins are going to help in the synthesis of this cell wall. So I told you that Golgi apparatus will help in the formation of hemicellulose and the pectin. But when it comes to the cellulose will be synthesized by the enzyme present in the plasma membrane. So that's it. That's also, there are so many diagrams that I have added. You can relate it, you can revise the things from these diagram. Okay, so ultimately cellulose is itself made up of what? Cellulose is made up of the glucose. It's a polymer of glucose, right? It's a straight chain, right? It's a straight chain polymer of glucose. Okay, bache? So see lignin, cellulose, hemicellulose. In the bacterial cell wall you can see peptidoglycans are present. In the fungal cell wall you can see the chitin is present. In plants, lignin, cellulose, hemicellulose and pectin. So these type of flowcharts are going to, you should even make that type of flowcharts, because then only you will be able to revise the things in a very easy manner. Okay? Okay. So the next thing that we need to discuss is endomembrane system, and even in NEET 2023 one question was there from this particular part, although the question was very easy. Even while solving the question, even I made a mistake. I was reading it, which organelles are the part of endomembrane system, but they were asking that which organelles are not the part of endomembrane system. Right, so I was just solving the paper, so I had that chance that I can, change my answer and I can tell you that this is not the correct answer, this is the incorrect answer, right? But while marking the OMR sheet, you need to be very accurate. So before marking the answers, you need to do what? You need to read the question twice. Okay, you need to read the question twice. Okay, done, bache? So now what are we going to discuss? We are going to talk about the endomembrane system. But before that, let's revise the cell wall from NCERT. In NCERT, it is given in a one paragraph only. So let's revise it from the NCERT. So any doubt? Bache, any doubt from this topic, cell wall? I hope everything is clear to you. So it's a non-living rigid structure called the cell wall. So in NCERT, it is clearly mentioned that overall cell wall is non-living, right? Overall cell wall is non-living. It is mentioned in the NCERT. Okay. Okay, so it forms the outer covering for the plasma membrane of fungi and plants. So it not only gives shape to the cell, but it protects the cell from the mechanical damage and infection. Now I told you this thing very clearly, that in the case of plants, right, it is the pectin, it is the pectin which will fight with that infection. Okay, so along with that, it also helps in cell to cell interaction and provide barrier to undesirable macromolecules, like I showed you that na, plasmodesmata, the continuous cytoplasmic connection. Now bache, when you talk about the algae, right, in the case of algae, the cell wall is also having cellulose, no doubt, but along with that galactins and mannans and minerals like calcium carbonate is present. This is important. In the algae, cellulose is there, but along with that, galactins, mannans and minerals like calcium carbonate are there. Galactins, mannans, mineral like calcium carbonates are there. But when you talk about other plant, it is just having the cellulose, hemicellulose, pectin, and even proteins are also present, right? Even some proteins are also cross-linked and they are present. So I mentioned cellulose, hemicellulose and pectin now, but proteins are also there. So this is what you need to take care. These minor details are very important for your NEET examination. So cell wall of a young plant, it is the primary, it is capable of growth, it will gradually, slowly it will diminish as the cell matures, and secondary cell wall will form the layers there. Okay bache, so middle lamella is a layer mainly made up of calcium pectate. It is going to, which holds or glues the different neighboring cells together. So it is very easy. Okay, it is very easy, right bache? So now when we talk about the endomembrane system, so see, although we have different organelles in a cell, but there are some organelles in which, you know, the function, they are functioning in a coordinated manner. What are they doing? They are functioning in a coordinated manner. Their function is all together, right? So yes, there is one trick for that. When it comes to the endomembrane system, we talk about the GERL complex. GERL complex. G stands for Golgi apparatus. ER stands from endoplasmic reticulum, right? Endoplasmic reticulum. And when it comes to the L is basically the lysosome, and along with that we are also having the vacuoles as a part of endomembrane system. Okay, we are even having the vacuoles as a part of endomembrane system. So you can check out this diagram, right? Endoplasmic reticulum, Golgi apparatus, lysosome is there, the vesicles are there, and even vacuole is there. This is a very good diagram. It's a beautiful diagram. It is going to explain everything, right? You can see this. So see how things are arranged. Like this is the endoplasmic reticulum. Some secretory vesicles are coming, right? Some transport vesicle rather. Some transport vesicles are coming from this endoplasmic reticulum. Now they are going to fuse, right? They are going to fuse with this Golgi apparatus. You can see this. It is going to fuse with this Golgi apparatus, right? Now in the Golgi apparatus, endoplasmic reticulum is making something. It is present in these transport vesicle. They are containing the secretions of the products of this endoplasmic reticulum. Now it will fuse with the Golgi apparatus. It will fuse with this side of the Golgi apparatus, and in the Golgi apparatus further modifications will be done. It's again an important question, right? The further modifications will be done. You can say that glycosylation, glycosidation, like let's say endoplasmic reticulum is making a protein. So that protein will be there in this transport vesicle. Now this transport vesicle will fuse with this Golgi apparatus, and here to this protein, right, the carbohydrate part will be attached. So it will become glycoprotein. So such type of modifications occur here, right? And if you talk about the smooth endoplasmic reticulum, it will form lipids that will also fuse here. Then it will further add carbohydrates if required. So glycolipids will form. So that glycolipids, glycoproteins, they will be formed, they will be modified here in the Golgi apparatus. And again, right, some secretory vesicles will be released from Golgi apparatus after all that changes. So they will go to their designed place, right? They will go to their destined place. Okay, and lysosomes are also made by this Golgi apparatus. The composition of lysosome, that is hydrolytic enzyme, is also made by it. Okay, and see, they are forming some vesicle which will go to their destined position. So this is how they work. So they work in a coordinated manner. So we are taking these organelles as the part of endomembrane system. This is what you need to remember. We will discuss it in detail, Gokul. Right now I'm just giving you the overview of this topic, endomembrane system. So this is how it works. Okay, this is how it works. So now let's talk about this endomembrane system. So the first question that you should know is the organelles which are the part of endomembrane system. Your endoplasmic reticulum, Golgi apparatus, lysosome is there, and then what do we have? We have the vacuole. What do we have? We have the vacuole. So now we need to understand these organelles individually. Now let's revise it from NCERT as well. So here you guys can see, while each of the membranous organelle, it is distinct, it is different in terms of structure and function, but many of these are considered together as endomembrane system because their functions are coordinated. Why? Because their functions are coordinated, right bache? So ER, Golgi complex, lysosome, vacuoles, they are present, right? So since the functions of mitochondria, chloroplast and peroxisome, they are not coordinated, so they are not the part of endomembrane system. So one question was there from this part in the NEET 2023. So now let's start with the endoplasmic reticulum. So tell me something about the endoplasmic reticulum. Whatever you know about the endoplasmic reticulum, just let me know in the chat section, everyone. Quick. What do you know about the endoplasmic reticulum? Just let me know in the chat section. Quick. Whatever you know. And bache, there is no need to spam here, please. मॉर्निंग में कौन स्पैम करता है? मॉर्निंग में या तो पढ़ने आते हैं लोग या तो आते ही नहीं है. Aditya is saying protein synthesis. Very good, Lekhna. RER is there, SCR is there. Very good, बच्चे. Juhi continues with the nuclear membrane, smooth and rough. Okay. Anything else? Anything else? Synthesized lipids. See, you need to be very specific. You are right when it comes to the endoplasmic reticulum. It is basically going to make the framework of the cell, right? We have two types. We have one is SER, smooth endoplasmic reticulum, and another is RER, that is rough endoplasmic reticulum. One is a smooth and another is a rough. So when it comes to the rough, endoplasmic reticulum is having different shapes. I will not discuss that now. I'll explain it. Just give me a minute. So when it comes to the smooth rough endoplasmic reticulum, its surface is rough because it will be having the ribosomes over it. It will be having the ribosomes over it. So obviously if it is having the ribosomes, because ribosomes are the protein factory, so rough endoplasmic reticulum is going to form the proteins. And when you talk about the smooth endoplasmic reticulum, its surface is smooth. It is not having these ribosomes over its surface. So it mainly plays role in the synthesis of lipid, right? It helps in the formation of lipids, in cholesterol formation. So this smooth endoplasmic reticulum, it even helps in the formation of the steroidal hormones, right? Even it helps in the formation of what? In the steroidal hormones. So this is what you need to remember. So with the advancement in the microscopy, with the help of electron microscopic studies, we were able to see their structures, right? So electron microscopic studies of eukaryotic cell, they reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm, and that is ER. So with the help of endoplasmic reticulum, with the help of electron microscopy, we are able to see the endoplasmic reticulum, right bache? We are able to see the endoplasmic reticulum, right bache? Right bache? So if you talk about this endoplasmic reticulum, again here also we talk about the different shapes. First of all, when the word is reticulum, reticulum in general means network. What is the meaning of reticulum? Network, right? Reticulum of nerve cells means network of nerve cells, right? What is the meaning of reticulum? Network. So endoplasmic reticulum is the word. So now you know that it is the organelle. It is having the membrane. It's a membrane bound organelle, right? What is it? It is a membrane bound organelle. Now here in this organelle, see, it is present in the cytoplasm, of course. So here in this organelle, it is having the membrane, so obviously it is doing that compartmentalization of cytoplasm. So it is the inner side and it is going to be the outer side, right? It is going to be the inner side, it is going to be the outer side. So we can say that there are two faces, right? Like see, ER divides the intracellular space into two distinct compartments. Luminal means inside ER, extraluminal means cytoplasmic compartment, right? So what do we have? We have two words here. Luminal means inside the endoplasmic reticulum, and then we have the extraluminal means cytoplasmic compartment, like you can mention it here. So this is the luminal inner side, right? And it is the extraluminal. It is the extraluminal cytoplasmic side. And bache, in endoplasmic reticulum also, it can have different shape. The tubules can be there, vesicles can be there, right? And it can be, you know, some sac like, tube like can be there. It can be it can have vesicles, right? It can have vesicles. Okay, and one more shape is there. Can you tell me about that? Can you tell me about that? Tell me, anyone. We have three things now. Tubules are there, tube like structures, no doubt. Vesicles, some rounded structures are there. One more shape is there. Can you tell me about that? Anyone in the class? Anyone in the class? Yes, very good, very good. Balapunuri, right? Very good, excellent. Cistern, right? We have the cistern, the sac like structure, the pouch like structure. Cistern, no Anuradha. Cistern, tubes, vesicle. Tube means like, you know, it can be branched or something like this. Some tube like structure, vesicle, some rounded structure. And when you talk about the cistern, some sac like structure, right? Having inner lumen, some sac like structure. Even such shapes you will even see in the Golgi apparatus. The cisternae, tubules, vesicles, such shapes you will even see in the case of what? In the case of your Golgi apparatus. So now, bache, you are right when you talk about the nucleus, right? See, bache, note down. We have some double membrane bound organelle. There are some organelles in the cell that are double membrane bound. There are some organelles in the cell which do not have membrane, no membrane bound, or you can say that membrane is absent. This is important. I'll tell you why. See, so basically what is happening when you talk about the double membrane bound organelle, you have the example of nucleus, you have the example of mitochondria, right, bache? And even you have the example of plastids. Are you getting my point? You have the example of nucleus, you have the example of mitochondria, you have the example of plastids. When you talk about the double membrane bound organelle, in such organelles you will be having outer membrane and the inner membrane. The outer membrane is there, inner membrane is there. And there are some no membrane bound organelles also, like ribosome. In the ribosome there is no membrane, right? And moreover, centriole or centriole in the case of animal cell, it is also a non-membrane bound organelle. So whatever is left, that is going to be single membrane bound, right? This is how you have to remember, Akhilesh. Double membrane bound is nucleus, mitochondria, plastid. And when you talk about the organelles in which the membrane is absent, ribosome is there, centriole is there. Ribosome is there, centriole is there. Okay, they do not have the membrane. So whatever is left, your endoplasmic reticulum, your Golgi apparatus, your lysosome, they all are having the single membrane, right, bache? They all are having the single membrane. So now when you talk about the nucleus, the outer membrane, right? So it is also a double membrane bound organelle, inner and outer. So outer membrane, right? So endoplasmic reticulum is continuation of this, right? Endoplasmic reticulum is the continuation of this outer nuclear membrane. It is the continuation of this outer nuclear membrane. And here you will see, near to the nucleus, here you guys are going to see some network of tubes like this. So if it is having the ribosome, then it will make the rough endoplasmic reticulum. If ribosome is not there, then it will be the smooth endoplasmic reticulum, right, bache? It will be the smooth endoplasmic reticulum. So let me show you this thing here. So here you people can see. So outer nuclear membrane, the continuation. See this part, rough endoplasmic reticulum, right? And here it is the smooth ER, smooth endoplasmic reticulum. And do you know, bache, what is the reason behind? See, in smooth endoplasmic reticulum, some, you know, ribophorins are absent. So these ribophorins, they help in the attachment of ribosome. So let me mention it here. Like in the rough endoplasmic reticulum, we have the ribophorins present, right? What do we have? We have the ribophorins. So because with the help of these ribophorins, the ribosomes are able to attach to this endoplasmic reticulum, right, bache? The ribosomes are able to attach to this endoplasmic reticulum. Are you getting my point? Are you getting my point? Okay, okay. So RER and SER, smooth endoplasmic reticulum and rough endoplasmic reticulum. Ribofurans present, with the help of that the ribosome can get attached to the endoplasmic reticulum, and here the ribofurans are absent, so it is smooth. Now bache, ribosome is there, obviously it will play the role in the protein synthesis. So the cells which are actively forming the proteins, they will be having so many rough endoplasmic reticulum, and when it comes to the SCR, as I said, in the synthesis of lipid it is going to play the role, in the formation of cholesterol, in the formation of steroidal hormones it will play the role. Okay, and I will add one more function of SCR here. But do you know liver plays the role in detoxification? Do you know that liver, it is the liver which plays the role in the detoxification. It is the liver which plays the role in the detoxification. Do you know about it? Do you know about it? Liver plays the role in the detoxification. Right? So this smooth endoplasmic reticulum is going to help in that. Right? Whatever the drug metabolism is there. So basically it helps in settling down that things. So now this is bache your homework. You are going to write the differences in, you will write the function of SCR and RER. It is very important. You have to write the function of SER and RER. You never know from which function they can ask you the question. And even it is going to help you out in long run. Even if you will join the MBBS, you need to know these details. So it is good to revise it now. It is good to revise it now properly. Right? Okay. So now let's have a look here. So here you can see the endoplasmic reticulum. Ribosomes are there. So obviously it is rough ER. Done. So if it is not there, RER. Okay. So RER is frequently observed in the cells, actively involved in protein synthesis and secretion. Right, bache? Right, bache? And when it comes to the smooth endoplasmic reticulum, what will they do? Synthesis of lipid. Right? So lipid-like steroidal hormones are synthesized in SCR. A very important function, bache. Okay, a very important function. Now the next is Golgi apparatus, and this Golgi apparatus in the case of plants we used to call it as dictyosome. Right? In the case of plants we used to call it as dictyosomes, the Golgi apparatus. In the case of plant, what we used to call it? We used to call it as a dictyosome. Now bache, Camillo Golgi in 1898. Okay, he observed these densely stained reticular structure near the nucleus. Again, you need to focus on each and every word. Reticular structure. Reticular word is related to the network. Right? It is related to the network. And yes, one more thing. In endoplasmic reticulum, I told you cisternae, tubules and vesicles are there. So you need to even check which like smooth endoplasmic reticulum. Majorly you will find it in the form of tubules or in the vesicles. Such type of points are also there, and you need to focus over there. You are going to revise it, you are going to note it down. Right, bache? See, bache, if you are getting up early in the morning, you are attending my lecture, right? You are listening to me. So whatever homework I am giving you, please write it down. Okay, if you are spending your time, if you are investing your time now in this lecture, so do not just waste your time, please. I can see some students are just talking to each other, they are just spamming. It is of no use. You can do it on other social media platform as well. You can do it on other social media platform as well, isn't it? So right now if you are attending a session, then be a good student now. You are investing your time, you are getting up early in the morning. So let's utilize that time in a better way. Let's complete that chapter properly. Done? Okay, so now the next part is the Golgi apparatus, and when the word is reticular, it means network. It means network. Okay, so again, here in the case of Golgi apparatus, we will be having some cisternae, you know, some even vesicles will also be there. So that is how we used to draw it. That is how we used to draw it, right? So let me show you this part. Right, again it is a network. If you will observe it properly now, some these cisternae, these pouch like structures are there. Again it is a network, and it is near to what? It is near to the nucleus. Here, see, it is the nucleus. Now bache, when you talk about this Golgi apparatus, see the endings are swelled up now. See, this is the only organelle. It is the only organelle having two faces. two different faces, and even you will see their function is also different. I'll mention it. It is the only organelle having two faces. Like if you talk about the face which is near to the nucleus, we used to call it as cis face, right? Even it is known as forming face, right? It is the cis face. It is also known as forming face. And when you talk about this side, it is the away from the nucleus. It is the trans phase, also known as maturing phase. Okay, trans phase, also known as maturing phase. Trans phase, also known as maturing phase. So yes, it is a organelle. It is a polar organelle. It is having two different phases. Even their function is different, but still, see, they are interconnected. Still they are interconnected. This is what you need to know. And moreover, wherever this Golgi apparatus is present here, this Golgi apparatus is present here. So the cytoplasm, so this region where this Golgi apparatus is present, just look at it, right? This region where this Golgi apparatus is present, in this cytoplasmic region, no other organelle will be there. Right, here in this region where this Golgi apparatus is present, no other organelle is there. So we used to call it as zone of exclusion. We used to call it as zone of exclusion. So it is the region of cytoplasm where your Golgi apparatus is present, and now here in this region no further organelle will be there. So that is why we used to call it as zone of exclusion, right? Right, zone of exclusion. Near to this nucleus, there will be the cis phase, forming phase, and this side there will be the trans phase, there will be the maturing phase. So now bache, what is going to happen here? You know na, you will be having the endoplasmic reticulum as well. So from the endoplasmic reticulum, some transporting vesicles will come. Endoplasmic reticulum is going to form the protein. Let's say it is rough endoplasmic reticulum. So this transporting vesicle will come and it will fuse with the membrane of this Golgi apparatus. Okay bache, Juhi, it will fuse with the membrane of this Golgi apparatus. Now again it is the cistern, matlab sac like structure. It is a sac like structure. Okay, what is it? It is a sac like structure. Inside there is the lumen. Inside what is there? There is the lumen, right bache? So transport vesicle is having something, it will enter here. Now the further modification will be done. Okay, like the word is glycosylation and glycosidation, right? Glycosylation and glycosidation will occur in the Golgi apparatus. Like glycolipids and glycoproteins and glycolipids will form here in the Golgi apparatus. Basically there will be the modification. There will be the modification. Are you getting my point? And even you know, this Golgi apparatus is also known as the traffic police of a cell because it helps in the sorting. It helps in the sorting of the things. It is also known as the traffic police of the cell because it helps in the sorting of the things. Right? It will tell the things that okay, you need to go to this part. Like let's say there is a protein, it will send that protein to the plasma membrane if plasma membrane needs it. Right? It will send the particular things to that their particular place. So it is just like the traffic police of the cell. It is going to guide it. It is going to put some markers that you need to go there, you need to go there, you're required there, you're required there, like this. Okay? Like this. Okay, yes Prasanna, even you can mention it. It is the convex phase, it is the concave phase. Very good. Are you getting it? So it is the traffic police of the cell. It helps in sorting of the things, sorting and arrangement, right? It helps in that. Getting my point? So from this transporting vesicles, the things will come here, the modification will occur, and finally when it is mature, so secretory vesicles will come from this side, from trans phase. Secretory vesicles will be released from this part, obviously, from this part basically, and they will go to their destined places. So this is how it works, right? This is how it works. And moreover bache, when you talk about the Golgi apparatus, as I said, it helps in the synthesis of cell wall, right? Even in the case of sperms, it forms the acrosome, right bache? And even it helps in the formation of the enzymes of lysosome. Do you remember lysosome, the suicidal bag containing hydrolytic enzymes? Right, lysosome, the suicidal bag containing hydrolytic enzyme. So it is going to form that too. So this is what you need to remember, the function of Golgi apparatus. Very important it is. So I want you people to write a note on the functions of Golgi apparatus. So here you people can see the Camilo Golgi. He first observed densely stained reticular structures near the nucleus, right? Later on they were named as Golgi bodies. So they are having flat disc shaped sacs or cistern, pouch like structure, right? So it is 0.5 micrometer to 1 micrometer in diameter, and they are stacked parallel to each other, right? They are stacked parallel to each other, you can see here. So there are, see, cisternae, varied number of cisternae are present in Golgi complex. Like you cannot be sure that there will be 2 only, 3 only. No, it can vary. It can vary, and they are stacked like this. They are arranged like this, like 1, 2, 3, like this. Okay, they are arranged. Okay, so Golgi cisternae are concentrically arranged near the nucleus with the distinct convex or concave phase. So convex, cis or the forming phase, concave, the trans phase, maturing phase, right? So both the phases are entirely different, but still they are interconnected. They are entirely different. They are totally different, but still they are interconnected, right? So packaging will be done here. It will be delivered to the targets, bache, right? So materials to be packed in the form of vesicles. So here you can see from ER it will fuse with cis phase. I have already explained it, and then it will move towards the maturing phase. Okay. Okay, so that is why they all are in close association. That is why they are the part of endomembrane system, because they are functioning in a coordinated manner. Okay, they are functioning in a coordinated manner. They are working together, bache. Done, bache? Okay. Okay, so glycosylation and glycosidation will be done here. So now we will talk about the lysosome, also known as suicidal bag, the organelle which shows polymorphism. Do you know that? It is the organelle that shows polymorphism, right? It shows polymorphism. So these are the suicidal bag. Why? Because they are fully loaded with the hydrolytic enzymes that can destroy the components of the cell, right? Lysosome, suicidal bags, right? So they are even, these enzymes are also synthesized by Golgi apparatus. So they are having all the hydrolases like carbohydrases, lipases. So it can destroy the component of the cell. So what are they? They are the membrane bound vesicular structure. They are also formed by the process of packaging in Golgi apparatus. During that packaging, when that vesicles will come out from that cisterna, so even the lysosome form. So what do they have? They have the hydrolytic enzyme. What do they have? They have the hydrolytic enzyme, right, bache? So now which hydrolytic enzymes are there? Hydrolases, acid hydrolases, of course. They are the lipases, proteases, carbohydrases. So they are active at the acidic pH. Like when the pH is basic, then these enzymes are not at all active. They are not going to harm anything. But when the pH is acidic, then at that time these enzymes will become functional. So lipases will destroy lipids, proteases will destroy proteins, carbohydrases will destroy the carbohydrates. So in that way, ultimately your cell is made up of that things only, so it can destroy the cell. Right, it can destroy the cell. And now, bache, the question here is that these enzymes are present within the lysosome, but still they do not affect the lysosome. It is doing nothing to the lysosome. Can you tell me why? These hydrolytic enzymes, they are present in the lysosome here, but still they are doing nothing to this lysosome. Can you tell me why? Can you tell me why? Why is it so? Anyone in the class? Yes, anyone in the class? Why? We have the lysosome fully loaded with the hydrolytic enzymes. Although they in the lysosome they are not active, right? But still they are not doing anything to the lysosome membrane. Why? Nope. But the composition of lysosome membrane is the answer here. The composition of lysosome membrane is little different. In the lysosome membrane, we have some compounds which are going to protect this lysosome from these enzymes. Okay? No. The composition. So now this is a question for you. This is again the homework. I want to see the answer in the chats in the comment section later, right? So what is present in the lysosome which is protecting it from these hydrolytic enzymes? This is what you need to tell, right? What exactly is that compound? This is what you need to mention. Okay, bache? This is what you need to mention. Clear? So now, as I said, that lysosome is the only organelle which is going to show what? Polymorphism. Can you explain how? First of all, bache, let me tell you, whenever there is any dead organelle present in the cell, so what is going to happen? This lysosome will fuse with that dead organelle. Let's say that is a dead organelle, right? So it will fuse with that, right? And what will it do? It will eat it. The enzymes which are present there is going to destroy it. And moreover, whenever there is the condition like this, like let's say this cell is not working well, right? And there is the need to destroy the cell. There is the need to damage the cell because the cell is not working well. Cell death is required. At that time also, the lysosome is going to release its content and it can destroy the entire cell. So it helps in the cell death also whenever required. So now I just mentioned that lysosome is the organelle which used to show the polymorphism. Can you tell me how? Like remember, there are different forms. Primary lysosome is there, secondary lysosome is there, and we are even having the tertiary lysosome, also known as residual lysosome, right? So when it is the primary lysosome, it is simple lysosome where enzymes are not at all active, right? Where enzymes are not at all active. And when we use the word secondary lysosome, it is even known as heterophagosome, right? It is even known as heterophagosome. So what is going to happen? This lysosome, it will fuse with the food vacuole, right? It will fuse with the food vacuole. So now it will fuse with the food vacuole. Obviously, here you have the hydrolytic enzyme, here you have the food vacuole, so it will help in the digestion there. So we can also call it as a digestive lysosome now. It is going to digest that particular food present there, right? It will help in that digestion of that food. Okay, so it is the digestive lysosome. Now after the digestion, whatever is remaining, right, in the lysosome, whatever is left, then it will become the residual lysosome, and then it will throw it out. Right, bache? So in that way it shows the different shapes, right? And even the function is also different. So that is why we used to say that lysosome is the organelle which used to show the polymorphism. And when that lysosome destroy the organelles or the cell, we used to call it as autophagosome. Auto means self death. Now it is going to destroy that cell completely. Okay, autophagosome. So it is very easy and you should revise it. It is not given in NCERT, but still the question used to come from this part. So you need to revise it. Okay, you need to revise it. But next is the vacuole. So in the case of vacuole, the very important point that you need to understand is that in the case of vacuole, there is a membrane, and we used to call that membrane as tonoplast. Right? There is a membrane in the case of vacuole, and we used to call that membrane as tonoplast. What is the name of that membrane? The name of that membrane is the tonoplast. So it is important, bache, please mark this word, tonoplast. So it's a membrane bound space in the cytoplasm. It can have water, sap, excretory product and other materials which are not useful for cell, and vacuole is bounded by a membrane called tonoplast. Done, bache? Tonoplast. Okay, so bache, when we compare the plant cell and the animal cell, you know that in the case of animal cell, the vacuoles are comparatively smaller in size. But when you talk about the plant cell, right, the vacuole is going to be very large, and because of that, nucleus is pushed to the one side. Right, so here this vacuole in the case of plant cell is very important, because when water used to come inside now, even it used to go in the vacuole, so it will even help in that elongation. And in the plant physiology, we will discuss the phenomena like, you know, plasmolysis, how the water will come. So everything we are going to discuss. So here, remember, the vacuole is also a membrane bound organelle, and the name of the membrane is tonoplast. Right, the name of the membrane is tonoplast. Even it is having the pumps as well, which will help in the active transport of ions. Right, it will even help in the active transport of ions. Are you getting my point? So you guys can see here that in the plant cells, vacuoles can occupy up to 90% of the volume of cell. It can occupy up to 90% of the volume of cell, bache. Right? So as I said, the tonoplast, it will facilitate the transport of number of ions even against the concentration gradient. So when the word is against the concentration gradient, means we are going to talk about the active transport here, isn't it? We'll be discussing the active transport here, isn't it, Rohit? We will be discussing the active transport here, right, bache? So their concentration is significantly higher in the vacuole than in the cytoplasm. So this is something very important. So basically the ions which will be actively transported, like in the case of vacuole, we can say that in the case of plant cell, we can say that the vacuoles will have more ions in comparison to the cytoplasm. There are some ions which are more in vacuole in comparison to the cytoplasm. Okay, so in tonoplast layer we are also having the pumps. So when we use the word pumps, means they are going to play role in the active transport. They are going to play role in the active transport. Exactly, Anuradha. They will help in the movement of things against the concentration gradient. Okay, so the name of the membrane is important. This line is important when it comes to the vacuole. Okay, so even in amoeba, which is a eukaryote, which is a eukaryotic cell, and it is a freshwater organism, it is having the contractile vacuole. It will help in maintaining the osmoregulation. It will help in maintaining the salt and water balance, right? Salt and water balance. Are you getting my point? Right? So in many cells, in Protista, food vacuoles are also there, which will engulf the food particle, which are going to engulf the food particle. So this is what you need to remember. So this is all about, right, bache? So this is all about your what? Okay, this is all about your, yes, endomembrane system. Okay, so today what we revised? Today we revised the cell wall and endomembrane system. So in the next class, that is going to be the last class, we will complete mitochondria, your plastids, ribosomes and cytoskeletal element. Okay, what is the main importance of bache? All the organelles are important because they are playing a specific role. So if you will look at these organelles individually likhna, so endoplasmic reticulum is making the proteins and lipids, again important for the working of the cell and of the entire body. Even the Golgi apparatus, that modification is important, right? The acrosome of the sperm is made by Golgi apparatus, again it is important. There are different roles. It helps in the synthesis of cell wall as well, right? Even the lysosome is important. When the cell is not functioning properly, we need to destroy that cell. Cell death is important. It is helping there. And vacuole is playing a very important role in the case of plants as well, and in maintaining the osmoregulation and digesting the food. So it is important. So do you have any other doubt, bache? And yes, how many of you have joined the Avengers batch? Yes, tell me. How many of you have joined the Avengers batch? See, if you have joined it, very good. Right, because classes are going to start from 25th of May and then there will be no delay. There will be no delay. We are not going to accept any further enrollment after 25th of May. The reason is we do not want your backlogs. We do not want students to have the backlogs. Right? Because of backlogs, you are not able to revise properly, and even we don't like it. But still, if you are thinking to join it, just go for it, because it is one of the best batches for the NEET preparation. The team is enthusiastic, right? We are experienced. We know what to deliver, what not to deliver. Right? There are many things. There are many topics which you need to understand, and for that you need to go beyond the NCERT. But you cannot read everything. Right? You should know the limits. So in less time, right, that quality content, that required content will be delivered by us. So we want you people to be the part of Avengers batch if you are a serious aspirant. If you are a serious aspirant. If you are not, then please no need to join our Avengers batch, because we want the students who are having that vision that yes, this time I am going to crack it. I am ready to do the hard work. Right? I do not have the time to waste. Whatever my teachers are going to tell me, I am going to follow that, and I have to follow that. Right? And bache, on YouTube you will get the quality content, no doubt. I am not going to teach this topic in a different way in the Avengers batch, but in that Avengers batch there will be a connect. I will be able to cater your doubts. I will be able to give you the homework. We can practice the questions there. Right? We can practice the questions there. You can check that recorded lectures right afterwards also. Let's say because of any reason, let's say you have missed one class, so obviously you will be able to check the recordings. But again, let me tell you, you're not allowed to miss any class. So be the part of this Avengers batch. You can check any of our video. You just need to go to the description box, click on the batch link here, bache, and you need to enter your mobile number and you have to continue with the email. So see, the coupon code is already applied, that is Avenger. You will get this batch in 9499 rupees. 9499 rupees, bache. So quality content, study material, revision, mock test, everything will be provided to you. And trust me, if you go for the offline coaching, you used to spend 1 lakh or 2 lakh rupees for such content only. Right? You are going to spend that much money, but now you are getting everything at this price. At this price. and it is not too much. Literally, it is not too much. Okay, so and even when you pay for something, now you have that, then you get that seriousness. Now I have paid for it. Right, I have to justify that amount, and even that automatically that seriousness will come out. And but YouTube is very, it used to distract as well. If you cannot even afford that, okay, fine, you can be the part of this batch. You can study from us. Obviously, we are going to teach you free here on YouTube as well. But again, that connect and everything is required. On YouTube, if you will check, you will find a very good content over YouTube, but still many students are not able to get selected because it is not just the content. You need to follow a routine. There should be a batch. You should know what to revise, what not to revise, because after few months of preparation, you will be baffled. You will not be able to realize that now I should go for the physics or for the chemistry or biology. How should I complete the syllabus, right? So all that things can occur. But when you are enrolled in a batch, there will be a discipline, there will be a decorum, right? So you will go in a flow. So that's what you people need to do. So be the part of this batch as soon as possible. Do let me know the homework in the comment section as well, and be consistent, be regular, bache. Okay, be consistent, be regular. Okay, so there is no need to miss the class. Consistency is the key. It is the consistency which is the key, and please go for that. So take care, bye-bye, thank you. So there will be one more class for the cell chapter, then we will complete the entire, right? We will complete the chapter then. Okay, so thank you so much everyone.